Good evening to one and all. Good evening, sir.
Yes.
This is Dr. A. Rajni, Head Department of Botany and Food and Nutrition, Rbvrr Women's College. We are started to the 2 days back the faculty exchange program. Now today also we start to the faculty exchange program. Sir name is Padala Tirupati, SRR Degree College, Botany Department. Sir, to start the session, nitrogen metabolism. Today also to continue the nitrogen metabolism. Okay, sir. Thank you.
Okay, very good evening, respected HOD Madam Garu, Dr. A. Rajni Madam Garu, Assistant Professor, Head Department of Botany, Food Science, Nutrition, Rbvrr Women's College, Autonomous, Hyderabad. Thank you very much for giving me this opportunity to teach and interact with our dynamic students in this night time. So, hearty welcome, my dear friends. This is Dr. Padala Tirupati, Assistant Professor, Department of Botany, SRR Government Arts and Science College, Autonomous, Karimnagar. In the program of Faculty Exchange Program, it is a wonderful program, exchange between two reputed institutions, two autonomous institutions from Hyderabad and SRR Government Arts and Science College, Autonomous, Karimnagar. So both the departments is going to interact with students by amazing ideas and going to conduct amazing programs. So in this regard, today I am going to explain about the biological nitrogen fixation. I hope the slide is visible.
Yes, sir.
Nitrogen fixation. Slide show కనబడుతుందమ్మా మీకు?
Yes, sir.
Slide show పెద్దగా కనబడుతుందా నిండుగా?
No, sir.
No. Okay. Now second slide is visible now, biological nitrogen fixation.
Yes, sir.
Okay, so we know that the nitrogen is a major component of all the living organism and also the molecules like proteins and amino acids and etc. Even though the magnesium, even though the important factor in the chlorophyll, the high amount of the nitrogen is available in atmosphere. But that is in molecular state. That is nothing but dinitrogenous gas. So high amount of the gas, that is nitrogen. 78% of the gas which is available in the atmosphere. Okay. But the source of cell nitrogen by way of its reduction to ammonia by biological agent is called biological nitrogen fixation. If it is available in the atmosphere, it is not usable form, but in the process of involvement of the biological agents like prokaryotes, these prokaryotes are useful to conversion of the atmospheric nitrogen into usable forms of the nitrogens. So in this way, nitrogen fixation by involvement of the organism is more important. Okay. So that's why this process we can call as biological nitrogen fixation. Okay, then the reduction of nitrogen to ammonia is catalyzed by key enzyme, one of the most important enzyme which is present in all prokaryotic organisms, that is nitrogenase enzyme. So the biological nitrogen fixation entirely depends upon the key enzyme, that is nitrogenase enzyme. Without nitrogenase enzyme, we cannot imagine about the biological nitrogen fixation. So without involvement of the nitrogenase, biological nitrogen fixation does not occur in all the entire organisms. The genes that code for the enzyme, which enzyme? Nitrogenase enzyme. So the genes which are responsible to code the nitrogenase enzyme, collectively we can call as NIP genes. The entire genes we can call as NIP genes. So NIP genes code for this dinitrogenase enzyme or nitrogenase enzyme. Am I clear? Then the next slide. Then modes of nitrogen fixation. We know that two modes of nitrogen fixation, biological nitrogen fixation, were observed in the entire world. That is the number one, symbiotic nitrogen fixation. Non-symbiotic nitrogen fixation, and second one is symbiotic nitrogen fixation. So these two modes were observed by involvement of the all organisms, or especially the prokaryotes. The prokaryotes held responsible either through fixation in symbiotic manner or asymbiotic manner. That is nothing but non-symbiotic nitrogen fixation. So the first one, non-symbiotic nitrogen fixation. This slide is visible. Non-symbiotic nitrogen fixation, my dear friends.
Yes, sir.
Okay. So the non-symbiotic nitrogen fixation is affected by those microorganisms which are living freely in environment without depend other organism. So these organism are these microbes which they can live, they can survive without depend on other organisms. So they can living freely in the atmosphere. So that's why the atmospheric free bacteria, the other living organisms, other prokaryotes, a large number of bacteria and some cyanobacteria are capable of non-symbiotic nitrogen fixation. The best example of these non-symbiotic nitrogen fixers are, number one, Azotobacter. It is a bacteria. Azomonas. It is a bacteria. Mycobacterium. It is a bacteria. Thiobacillus. Bacteria. Clostridium. It is also bacteria. Klebsiella. These are all the bacteria. These microorganisms, these prokaryotic organisms which are related to bacteria, which are all the best example of bacteria which are used to fixation of the nitrogen through asymbiotic manner or non-symbiotic nitrogen fixation. Am I clear? And the Azotobacter species, it is an aerobic. It is aerobic. And Clostridium bacteria, it is an anaerobic bacteria. So the non-symbiotic bacteria, which is also maybe the aerobic bacteria or maybe the anaerobic bacteria. What do you understand about the aerobic bacteria or anaerobic bacteria? Because without using of the oxygen or without involvement of the oxygen, the bacteria can capable to fixation of the nitrogen from the atmosphere without using of without depending on any other organism. So that's why the bacteria we can call as anaerobic bacteria. The best example of anaerobic bacteria, that is Clostridium. And the aerobic bacteria. Aerobic bacteria means the fixation of the nitrogen if it is available, if oxygen is present or with using of the oxygen, the bacteria can ability to fix the nitrogen from the atmosphere. That the bacteria we can call as aerobic bacteria. The best example of aerobic bacteria of the non-symbiotic nitrogen fixer, that is Azotobacter. The main nitrogen fixing free living bacteria. These two are free living bacteria. Okay, that means non-symbiotic bacteria. And some of the cyanobacteria members also. Cyanobacteria means blue-green algae members. Cyanobacteria means cyanophyceae algae members. Okay, these cyanophyceae members are cyanobacteria. Cyanobacteria may fix 10 times as much nitrogen as the other free living bacteria. They are mainly responsible for maintaining the fertility and productivity of rice field. The cyanobacteria members of the blue-green algae members, they are lived in the aquatic manner. So that's why in all rice fields, whenever we are cultivated the rice, in that rice field, the most suitable blue-green algae member, most suitable for the fixation of the nitrogen in the free living manner or non-symbiotic manner, that is cyanobacteria. And the Anabina and Nostoc are good example of the nitrogen fixing bacteria. Yesterday we are discussed, the Anabina and Nostoc. These two are the best example for the non-symbiotic nitrogen fixers, which belongs to blue-green algae or cyanophyceae members or cyanobacteria. And remember, the Anabina and Nostoc, these two are fixation of the nitrogen from the atmosphere in non-symbiotic manner with the help of heterocyst. Yesterday, we are discussed the nitrogen fixation depends upon the harmogonia. Harmogonia is very little amount. Harmogonia is suitable for the asexual reproduction, but not in using of the fixation of the nitrogen. But heterocysts are most suitable for the fixation of the nitrogen from the atmosphere, and the heterocyst spot is a non-chlorophyllous spot. Only the yellow-orange pigments are the carotenoids are present in the heterocyst. So that's why the heterocyst cell is compared to other trichome cells or other trichomes, other cells of the nostoc and anabina is quite differ. Why? Because chlorophyll is absent in heterocyst cell of the anabina and nostoc of the trichomes. And this is most important point, my dear friends. I hope slide. One second. The slide is visible.
Yes, sir.
Okay. Then the second one is associative symbiotic nitrogen fixation. Associative symbiotic nitrogen fixation, which means the organisms, they are associated with the different parts of the plants, maybe the roots of the grasses and cereal plants of the leguminaceae family members or Fabaceae family members. These bacteria grow in rhizosphere in close contact with the roots. Rhizosphere means the atmosphere of the surrounding of the root, we can call as rhizosphere. And sometimes invade the outer cortical regions of the roots and fix the nitrogen. So these bacteria are held responsible for the production of the or induction of the induced root nodules in the roots of the leguminous plants. The best example of the bacteria are the symbiotic manner nitrogen fixation, that is azospirillum, Pseudomonas, Enterobacter, and Bacillus. Okay. Then third one is symbiotic nitrogen fixation. Second one is associate symbiotic. Are you able to follow, my friends? Are you able to follow my class, my dear friends?
Yes, sir.
First one is non-symbiotic nitrogen fixation. Second one is associative symbiotic nitrogen fixation. And third one is symbiotic nitrogen fixation. So associative symbiotic nitrogen fixation without formation of the root nodules or without induction of the root nodules. Remember, my dear friends, the symbiotic nitrogen fixation, the third one is through root nodule formation in legumes. It is purely symbiotic nitrogen fixation. Okay. And the best example of the plants responsible for the formation of the root nodules in the rhizosphere, in the roots, the best examples are soya beans, glycinax, clover, alfalfa beans, peas, and some of the other leguminaceae croplands. And the bacteria being this symbiotic manner, that is Rhizobium leguminosarum, Bradyrhizobium, and Cynorhizobium or Cynorhizobium and Mesorhizobium, finally Azorhizobium. So these rhizobium bacteria are the rod-shaped bacteria. We know that the shape of the bacteria is quite different. Each and first one is what we can say the spherical type of the bacteria. That is what we can say that coccus or cocci, and rod-shaped bacteria that we can call as bacilli or bacillus. You know, so that's why these bacteria we can call as the rod-shaped bacteria, bacilli or bacillus. So these bacteria are gram negative. We know that Christian Gram has clearly shown about the what we can say the gram, Christian Gram, that we can say that the how to separate, how to identify, how to distinguish two different type of the bacteria, whether the gram negative bacteria or gram positive bacteria, by the using of the staining method or gram stain method. We can call it as gram stain method. In this method, the purple color, the bacteria cell wall and plasma membrane is adopting or the shown the purple color, that we can call as gram positive bacteria. If the bacteria cannot show the purple color, that bacteria we can call as gram-negative bacteria. Gram-negative bacteria. See here, our Rhizobium, Leguminosarum, Bradyrhizobium, these bacteria are gram-negative motile rod-shaped bacteria. Motile means the bacteria can have the flagellas. Okay, and here, the infection of the roots of a leguminous plants by these genera leads to the formation of root nodules that are able to convert atmospheric nitrogen to ammonia by nitrogen fixing mechanism. Remember, my dear friends, the bacteria are attracted by these bacteria, the entire these type of bacteria which are abundant present in this soil and which is very nearer to the roots of the leguminous plants and other plants. Whenever the leguminous plant has released a special type of sugars which are attracts these bacteria, and also the flavonoids like the lectin proteins also released by the microorganisms along with the plant root sphere or root environment, which are attracted to very nearer to the root part of the plants. And these bacteria slowly enter into the roots through root hairs. The root hairs are going to enter into the curling. Whenever the bacteria are entered into the the host plants, that is leguminous plants or leguminaceae plant or legume plant, the root hair is going to curl, and the curling part area we can call as shepherd crooks. Okay, then whenever the infection thread, the bacteria are reaches the the surface of the root hair, the infection thread is formed, then automatically bacteria are enter into the root hair, from root hair to the cortical region of the root. It is slowly which they are reaches the part of the internal part of the root, that is the cortical region. Here, the azorhizobium develops nodule on roots as well on the stem also. Generally, we are discussing about the Rhizobium leguminosum, which is held responsible for the formation or induction of the nodules in roots only. Here, the azorhizobium bacteria, which held responsible to develop the nodules on roots as well as stem also. This is most important, my dear friends. And here, the legume rhizobium nitrogen fixation is of considerable agricultural significance as it leads to a greater quantitative enhancement of the combined nitrogen in the soil. Whenever the bacteria which are reached the cortical region of the root, then automatically induction of the nodules. This nodule is held responsible for the fixation of the nitrogen from the atmosphere. So automatically the nitrogen quantity is rigorously increased inside the soil. Okay, that is the most important point. And about 90% of the all legume species become nodulated. Majority, 90%, more than all leguminous crop plants, which they can show the nodules. So that's why whenever the farmer or agriculturist are cultivated the crop plants related to leguminaceae or the Fabaceae family members, then automatically the soil is going to knowingly or unknowingly, whenever the farmers or agriculturists are the illiterates, even though illiterates, they are cultivated the crop plants related to leguminaceae crop plants, then the soil fertility is increased or decreased? Come on, tell me, my dear friends. Soil fertility is increased or decreased? Come on, anybody can interact, anybody can respond. Soil fertility is increased. Why? Because the nitrogen quantity or percentage of nitrogen is increased inside the root nodule, then automatically root nodules are also in the internal part or inside the soil, then automatically the soil fertility is increased. And remember, the more than 90% of the leguminous crop plants, they can show the root nodules. Okay. Then symbiotic nitrogen fixation. In previous slide, what we can say that azorhizobium. Then here the rhizobium strains, rhizobium leguminosum. The rhizobium strains are highly specific to legume species only, legume species. Some of the bacteria, non-leguminous crop plants also can ability to induce or production or formation of the root nodules. Okay, so that we can say that we can call as non-leguminous root nodules. But here the rhizobium strains are highly specific to the leguminous crop plants only, leguminous species only. A group of rhizobium strains capable of infecting a group of related legume is referred as a cross inoculation group. Before the bacterium can fix the nitrogen, they must establish themselves in the root cortical cells of the host plant, ultimately forming root nodules. As we can say that already in previous slide, whenever the bacterial strains reaches the cortical region of the root, then automatically the cortical region is going to expanded by the cell growth of the root nodular region. Then automatically root nodule is going to expand the size. Okay, expand the size. Automatically we can absorb the root nodules on the surface of the roots. Then here there are certain non-leguminous crop plants, although formation of the root nodules. What are they? Leguminous crop plants, we know that. Pisum sativum, Glycinax, Arachis hypogaea. Okay. The Phaseolus mangu, Phaseolus aureus. Okay. Vigna unguiculata. These are the related to leguminous crop plants. But non-leguminous crop plants also can ability to produce the nodules. The best example here, the plants are alder, that is alnus species, alnus. ఈ ఆల్నస్ అనేటువంటి జాతులు మనకు లెగ్యుమినేసి కుటుంబానికి చెందినటువంటివి కాదు. That we can say that non-leguminous crop plants. And the other, along with the alnus species, the Frankia species also, Frankia. Okay. So the temperate regions of the temperate region means a colder region. Okay. So tropical region means hot condition or very high temperature area we can call as tropical region. Here the temperate region means the cold region. The cold region area, the alnus species can ability to form root nodules. And even though non-leguminous crop plants, example Frankia also. Frankia is also ability to formation of the root nodules by the actinomyces bacteria. Actinomyces bacteria ఏం చేస్తాయంటే ఈ ఫ్రాంకియాలో కానీ ఆల్నస్ జాతుల్లో కానీ వేరుబుడిపలు ఏర్పడడంలో ప్రోత్సహించడం అనేటువంటిది జరుగుతుంది. Are you follow my class, my dear friends? Okay na?
Yes.
Then see here, my dear friends, even though the other leguminous plants that have root nodules are casuarina, sarugudu mokkal antam, casuarina, sarugudu, and the in the tropics and hippo, that is the buckthorn, which colonizes sand dunes and gravel soils. And without nodulation, in previous slides, what we have discussed, nitrogen fixation through formation of the root nodules. Here, without formation of root nodules, in the symbiotic, the mode of symbiotic nitrogen fixation, we are observed in the some of the organisms. See here, the non-nodulation nitrogen fixing associations are formed between Cyanophyceae and various plants. And even though the bacteria, Azospirillum and other plants. Okay. The cyanobacterium, that is Anabina azolla, which belongs to the pteridophyta plants or fern plants. It is an aquatic plant, hydrophytic plant, fern plant, azolla. So Anabina azolle, it is a cyanobacterium. It is a blue-green algae member, which is held responsible fixation of the nitrogen in the లీఫ్ ఆఫ్ ది అజోల్లా ప్లాంట్. అజోల్లా యొక్క పత్రంలో నత్రజని స్థాపన చేస్తాయి. అజోల్లా అనేటువంటిది ఫెర్న్ చెందిన మొక్క, అంటే టెరిడోఫైటాకు చెందినటువంటి మొక్క. ఓకే, సో టెరిడోఫైటాకు చెందిన మొక్కల్లో మనకు ఈ అనాబీనా అనేటువంటి నీలి ఆకుపచ్చ శైవలం సహజీవనం చేస్తుంది, సింబయాటిక్ మేనర్లో నత్రజని స్థాపన చేస్తాయి. సో దట్స్ వై ది అజోలా is best suitable for the rice fields growing very rapidly, and automatically the rice field is very fertile. Why? Because by involvement of the fixation with the nitrogen in the azolla leaves, azolla culture is most important. And nostoc is found in the stem of Gunira manicata or Gunira macrophylla, Gunira manicata or Gunira macrophylla. Okay, Gunira manicata and Gunira macrophylla. Okay, and even though the Azotobacter paspali develops colonies below mucilaginous root sheath of the Paspalum notatum. ఈ పాస్పాలం నోటేటం అనేటువంటి మొక్క యొక్క ప్రత్యేకించి వాటి లోపల ఏది మనకు మ్యూసిలేజ్ రూట్స్ అనేటువంటి ఉంటాయి. అంటే వాటిని మనం ఏమని పిలుస్తామంటే ప్రత్యేకించి ఈ శ్లేష్మంతో కూడినటువంటి ప్రత్యేకమైనటువంటి వేళ్ళు ఉంటాయి అనమాట. అంటే జిగటగా ఉండేటువంటి రూట్స్ ఉంటాయి అనమాట. ఇన్ దట్ ఏరియా ఇన్ దట్ కాలనీస్ ది అజటోబాక్టర్ పాస్పాలి ఎక్కువ వేగంగా డెవలప్ అయ్యి మనకు నత్రజని స్థాపన చేయడం అనేటువంటి జరుగుతుంది. And the Spirillum notatum, it is another type of bacteria which lives associated in around the roots of the digitaria and sorghum, maize. See, my dear friends, sorghum, maize, these belongs to the Poaceae family members, you know, graminae, grass plants, గడ్డి జాతికి సంబంధించినటువంటి మొక్కల్లో ఇవి మనకు ఉండడం అనేటువంటిది జరుగుతుంది. ఓకే, digitaria and sorghum along with maize. You know maize, కార్న్ ప్లాంట్స్, స్వీట్ కార్న్ అంటాం కదా, అవే అనమాట. సోర్గం, జవర్. ఈ సోర్గంను మనం ఏమంటామంటే గ్రేట్ మిల్లెట్ అంటాం మనం, గ్రేట్ మిల్లెట్, సోర్గం, జొన్న. దెన్ in association of rhizobium with leguminous roots and formation of the nodules seem to be obligatory for fixation of the nitrogen. మనకు ఏదైతే ఈ వేరుబుడిపేలు ఏర్పడడం అనేటువంటిది అబ్లిగేటరీ మీన్స్ ఖచ్చితంగా మనకు వేరుబుడిపెల్లో ఏర్పడడం ద్వారా మనకు నత్రజని స్థాపన చేయడం అనేటువంటిది ఈ రైజోబియం లెగ్యుమినేసారం అనేటువంటిది మనకు అర్థం చేసుకోవచ్చు. అంటే లెగ్యుమినేసి కుటుంబానికి సంబంధించినటువంటి మొక్కల్లో వేరుబుడిపెలు ఏర్పడడం ద్వారా మాత్రమే నత్రజని స్థాపన చేస్తే రైజోబియం లెగ్యుమినేసారం అనేటువంటి బ్యాక్టీరియా, గ్రామ్ నెగటివ్ బ్యాక్టీరియా, బ్యాసిలస్ స్పీసీస్ అండ్ రాడ్ షేప్ బ్యాక్టీరియా. ద స్టేజెస్ involved in root nodules formation include the following. See here, these are the stages. అంటే రూట్ నాడ్యూల్ ఫార్మేషన్ కి సంబంధించినటువంటి దశలు మనం ఇప్పటి నుంచి మనం చెప్పుకుంటూ పోతున్నాం అన్నమాట. How to formation of the root nodule. See here, the first step of the formation of the root nodule, that is recognition and attachment of the bacteria to the host plant root or the rhizosphere. అంటే వేరు యొక్క ప్రాంతంలో, భూమి లోపలి పొరల్లో, సాయిల్లో, నేలలో, మృత్తికలో రూట్ యొక్క వేరు పైన అంటే బాహ్య చర్మం పైన మూలకేశానికి మనకి బ్యాక్టీరియాస్ ఏ విధంగా అనుసంధానించబడతాయి అనేటువంటిది ఫస్ట్ వన్ గుర్తించబడాలి, అతకబడాలి. ఏ విధంగా చూడండి. రిమెంబర్ మై డియర్ ఫ్రెండ్స్, ది రూట్ ప్లాంట్స్ ఆర్ హోస్ట్ ప్లాంట్స్, ది ఆర్గానిక్ మెటబోలైట్స్ సెక్రెటెడ్ by the roots of the legume plants. Then the rhizobia migrate towards the grow inside the rhizosphere and built up to high population density or colonies of the rhizobium leguminosaurum. And my dear friends, remember these are considered as the flavonoid signals that are represent the plant organic metabolites, lead to the exchange of the recognition signals that is exchange or the attracting specific rhizobial species to specific legume root hairs. Here, my dear friends, the rhizobium and bradyrhizobium possesses a specific adhesion protein called ricardensin or on their surface. Then ricardensin is a calcium binding protein. Ricardensin is a calcium binding protein and binds calcium complexes on the surface of the root hair. Here, lectins are the proteins, my dear friends. Lectins are proteins. Lectins and carbohydrates containing proteins. Lectins are carbohydrate containing proteins also contribute in rhizobium legume attachments. So lectins are most important. Here, the ricardensine and lectins most essential for the recognition and adhesion. That means attachment of the bacteria on the surface of the root or root hair. Then penetration and travel. తర్వాత ఏం కావాలి? బ్యాక్టీరియా లోపలికి చొరబడాలి. ఎక్కడికి? వేరు యొక్క మూలకేశం లోపలికి చొరబడాలి. So after attachment on the root surface or root hair surface, then automatically root hair going to curls as a result of action substances excreted by rhizobium species. These substances are called nod factor. What we can say that? Nod factor. Nod factor has released by root hair or rhizobium species. Sorry, rhizobium species. Rhizobium species. Then after curling of the root hair, whenever the root hair is going to curl or అంటే వేరు యొక్క మూలకేశము వంకీని తిరుగుతుందన్నమాట. అర్థమవుతుందా? అంటే ఒక కొక్కె మాదిరిగా తిరుగుతుందన్నమాట. Then curling of the root hair, the bacteria penetrate and enter into the root hair and induce the plant to develop a cellulosic tube called infection thread. ఎప్పుడైతే బ్యాక్టీరియా వేరు యొక్క మూలకేశంలోకి చొరబడిందో ఆ చొరబడ్డ తర్వాత ఇమీడియట్ గా సెల్యూల్స్ తో కూడి ఉండేటువంటి ఒక నాళాన్ని అది ఏర్పరుస్తుంది. దీన్ని మనం ఏమంటే పిలుస్తామంటే ఇన్ఫెక్షన్ థ్రెడ్ అని పిలుస్తాం. ఇన్ఫెక్షన్ థ్రెడ్. ఈ ఇన్ఫెక్షన్ థ్రెడ్ then extends inwards to the root hair. Then whenever it is reaches the cortical region, see here my dear friends, whenever it reaches the cortical region of the root, then automatically the nod factors now stimulate root cells. Root cell is going to division. Cell division is occur to enhance or extend of the nodule or enlargement of the nodule, eventually leading the development of the root nodule by the involvement of the cell division, by the involvement of the nod factor, which is released by rhizobium bacterium. Am I clear? Then bacteroid. What is bacteroid? Bacteroid is the spherical shape of the enlargement space of the root nodule. We can call it as bacteroid. Here, the bacteria are released from the infection thread into the host cell cytoplasm. They get transformed into swollen, irregular shaped branched structure called bacteroid. Irregular branched structure called bacteroids. The bacteroid becomes surrounded singly or a small group by plant derived membrane called peribacterioid membrane to form structure called symbiosome. It is also we can call it the structure called as symbiosome. These symbiosomes are the sites of the nitrogen fixation. Where nitrogen fixation is going to fix? Nitrogen fixation takes place in symbiosome. Okay, symbiosome. Then symbiosome secretes a hormone which enables the production of leg hemoglobin. Remember my dear friends, leg hemoglobin is the oxygen scavenger. It is absorbs oxygen inside the bacterioid zone or symbiosome area. Why? Because the nitrogenase enzyme, which is only active in anaerobic condition. If oxygen is present, nitrogenase enzyme action is going to decrease. Very sensitive to the oxygen. Nitrogenase enzyme very sensitive to the oxygen. So that's why here the leg hemoglobin has released by host plant. Then remember my dear friends, leg hemoglobin, it is a globin protein which is released by host plant, leguminous plants. Then nitrogenase enzyme which is released by the rhizobium bacteria. Rhizobium bacteria present nitrogenase enzyme, the entire mechanism of the NIP genes, controlled by NIP genes. Okay. See, my dear friends, whenever the bacteria is going to enlarge due to the involvement of the cell division, that area is going to enlarge area, the nitrogen fixation area we can call as symbiosome. In the symbiosome, nitrogen fixation takes place by activation by the mechanism of the nitrogenase enzyme. But if oxygen is present inside the symbiosome, nitrogenase enzyme does not work. If nitrogenase enzyme works properly, we can absorb the leg hemoglobin, catch the oxygen. If the oxygen is present inside the bacteroid or the symbiosome, then leg hemoglobin catch the oxygen without disturbance of the nitrogen fixation. So that's why the entire mechanism we can call it a symbiotic process or symbiotic nitrogen fixation inside the root nodule. So root nodule formation and fixation of the nitrogen by bacteria, which is held responsible. Then automatically the host plant or leguminous crop plant, which is released by leg hemoglobin, it is a globin protein, which responsibility to catch the oxygen, minimum oxygen, even though a little amount of the oxygen is available inside the rhizobium, bacteroid or symbiosome, the leg hemoglobin catch the oxygen. So that's why whenever we are going to take the section of the bacteroid or symbiosome, that looks like a light pinkish color. Okay, మీరు ఎప్పుడైనా వెళ్ళి చూసారా? మీరు ఎప్పుడైనా ఆ రూట్ నాడ్యూల్ ను మీరు ఎప్పుడైనా కట్ చేసి చూసారా? If possible, whenever you observe the leguminous crop plants like Arachis hypogaea, okay, groundnut plants or soybean plants or glycine max or even though Dolichos labella, చిక్కుడు, వీటికి సంబంధించిన వేర్లను మీరు ఎప్పుడైనా తీసి చూసినప్పుడు ఆ రూట్ నాడ్యూల్స్ మీరు కనబడతాయి. ఆ రూట్ నాడ్యూల్స్ మీరు కట్ చేసి చూస్తే లేదా ఇలా చుంచి చూస్తే మనకు లైట్ పింక్ కలర్ కనబడుతుంది. బై బికాస్ the host plant released leg hemoglobin. So leg hemoglobin, due to the presence of the leg hemoglobin, the bacteroid or symbiosome looks like a light pinkish color. The main function of this leg hemoglobin is to catch the oxygen. Okay. For without destruction or without disturbance of the fixation of the nitrogen from the atmosphere. ఎందుకు అది ఆక్సిజన్ క్యాచ్ చేయాలి? ఎనరోబిక్ కండిషన్స్ ఉండే విధంగా. ఎనరోబిక్ కండిషన్స్ ఎందుకు అవసరం? వై ఎనరోబిక్ కండిషన్స్ ఆర్ నీడెడ్? నైట్రోజనేజ్ ఎంజైమ్ ఓన్లీ activated or action mode without oxygen or anaerobic condition only. So that's why oxygen is catched by the leg hemoglobin. It is released by host plant. Okay. So this is about the biological nitrogen fixation, my dear friends. I will stop here. Regarding of today's class, if you have any doubts, please ask me. I will clarify your doubts. మీకు దీనికి సంబంధించి ఏదైనా డౌట్స్ ఉంటే అడగండి. ఎనీబడి ఎల్స్?
ఏమైనా డౌట్స్ ఉంటే అడగండి సార్, క్లారిటీగా చెప్తారు.
నత్రజనికి సంబంధించిన జీవ నత్రజని స్థాపనకు సంబంధించి biological nitrogen fixation కు సంబంధించి it is the most important in your examination, any type of examination, competitive examination also. Without this biological nitrogen fixation question, we can't expect any UG, PG, research and PhD level, any level, and any botany, plant physiology. This is most important topic. Okay. So if